also a metal piece sticking out a little bit from the porch of the vehicle. Is that also an instrument, or is that a piece of the vehicle? Next to the slurp gun?
That is a temperature probe.
Ah.
The thing with the black ball on it to the left of the slurp?
Yes.
Yeah, that's our onboard temperature probe.
Got it. Okay. And then, Dan, I always like hearing how many. I feel like ROVs are like silent ninjas. They always have knives hidden everywhere. Are there any like knives visible for cutting off line if you you have run into line recently, but if you run into line?
Yeah, we have just to the right of the slurp, you see a yellow monkey fist, and then you kind of see a little black and red monkey fist. And those are attached to, you can see down on the grating there, there's a couple knives tie wrapped. One is kind of like a carpet knife, and the other one is a IKEA steak knife, which really cuts line under tension well. It's a serrated, both have a serrated blade on them. We actually used both of our knives not long ago. We discovered an old crab trap and got tangled up in it. Spent quite a while getting ourselves untangled, and we had to cut 2 lines to get ourselves free. Broke the first knife.
Wow, so that's why you have multiple knives.
Yeah, always carry 2. In fact, I'm going to hot up the manipulator so it doesn't, I don't know if Jess has done that yet. I'm under the philosophy that if the hydraulics are flowing in it, it's less likely to take on seawater. Yeah, so what we do with the T4s, they're live when they go off the deck, all the way down and all the way back up.
Is there any negative to that? Seems like if not,
They actually have to be, because they the seals in the rotary joints are pressure energized, so they need a delta across the vane seal to kind of pop in, if you will. So if you don't turn them on, it drifts around and gets stuck outside of the vehicle. The downside can be if you have a leak, then
you got it on for longer than you need to.
Yeah, you can lose your comp oil on the way down.
Yeah.
We have all the comps have sensors on them, so we know what the.
Yeah.
What are they? Have they strafed the gauges at all? Do they have any data there on the gauges?
Yeah, they got some data.
I did a 300. Curious to know what's in the res, because I just strafe them real quick. You don't have to write it down.
I think he's got UTC time on here. Yeah. 600. So no, it needs to do.
You can just cycle through and we'll have a look. 2 preset, 3 preset. That's the primary. Good, good. Good.
Oh, wrong one.
Really? Keep an eye on that one. What was the last reading for that guy?
Looking. 10. Oh wait, is this 1824?
Yeah, they haven't taken any readings
They haven't
yet.
taken any.
Let's
It
do
looked like it was right, but
I forgot to make my new sheet. Should take 30 seconds to check the gauges, not 3 minutes.
you changed all the presets?
No, you just go through all the presets and write down a number.
Oh.
Those, and you can use 2 lines on there, please.
Okay.
Because
Okay.
Kim and I can't read your young man's writing.
Yeah.
You just wait. Someday you'll be. I can't see that, what you wrote there.
Playing trombone with the paper.
20/20 vision till I was about 45. So really tragic. I wonder why she doesn't have it pegged. What are you doing here, Jess? Got auto heading on it. All right, come on.
I missed it when we were there, but we passed through the oxygen minimum zone about actually a while ago now. Our oxygen went down to about 3% saturation, 2 to 3, and now we're back up again, currently at 6.5% oxygen saturation.
I'm going to bump up the jam a little bit here. I have a date with a whale fall.
Do have a date with the whale fall. I like that. She's been there waiting. I think. Shouldn't go anywhere.
So you expect to find zombie worms there, do you?
Oh, I hope so. Yeah. We were talking about it a little earlier. When we came upon it last year, it already had Ossidax, the bone-eating worms, in the bones, but there was also a veritable carpet of worms kind of embedded and nestled in the blubber that had melted off the skeleton into the sediments below. And we don't know whether those worms will still be there. So yeah, some worms, yes, some worms, maybe they will be gone. It'll be really interesting to see just how much has changed in a year and how much is still left behind.
We just got to see one up in British Columbia that they've been studying for, what, 10 years?
They've been studying it for 10, and I think it was down for another 10 before that.
Wow, I didn't know they could last 20 years. Holy cow.
Wasn't much less.
So how much? Oh, it's okay. Were there still like bone parts, or
yeah,
what remained?
We counted how many vertebrae? So all the vertebrae and the jawbones were still there.
The ribs were mostly gone, I think, right?
Yeah, I didn't see any ribs.
Interesting. I'm curious to know, maybe this time with more of the tissue gone, if we'll be able to tell what species of whale that was that we're going to go to here.
Oh, they still don't know what kind of whale it was. Huh?
I think we narrowed it down pretty far. I don't think we got Oh, I don't think we got down to a species. We definitely knew it was a baleen whale, and I think we thought that it was. I'd have actually no. I'm not gonna say. I have to look at my notes to check. Kirsten Lindquist is on the chat, and she corrected me. The oxygen saturation actually went down to 0.5% of saturation when we were there in the oxygen minimum zone. So much lower than I spotted it at with 3%. I'm excited too for this first part of the dive where we're going to go to the octocone. We really came upon that just in the last, I think, hour or so of our dive when we were here last year. And we thought, based on the way that the feature looked, that there would be octopuses there, just like we saw octopuses at the Octopus Garden and Octo Spa, but we really weren't sure. We don't know yet why we found octopuses where they were, you know, what are the conditions. And so we ascended this cone of some volcanic origin and saw a lot of really neat pillow lava and a lot of beautiful stalked crinoids. And then right at the top of the octocone, we came upon more octopuses brooding in the crevices near what looked to be the same sort of warm water seeping out from the rocks. So, oh, I'm excited. We actually get some time to explore and see if there are more octopuses and kind of what the extent is on that cone. I wouldn't mind if we stumble upon a sponge too, but I know that's, you know, that's an Amanda thing. We saw some great sponges yesterday.
Can you check the Herc hydraulic page? I might have turned something on inadvertently. Herc hydraulics on the left. The encoder was on a funny button.
It looks good.
None of those are, just hit 0 on all the valves and stuff. Yeah, thanks.
Yeah, it was all, I think 0 anyway.
Amanda, you'd be glad to know that we even smelled your some sponge samples yesterday like you did last year. Oh, and this is Taylor Ann, by the way. I'm breaking for Mary. She's at dinner.
Oh my gosh, you remembered. Hi, Taylor Ann. I was pretty sure it was you, and I heard your name mentioned the other day.
Yeah,
oh, were there any notable or noteworthy smelling sponges?
It just smelled fishy, like seawater. But you know, I'm no expert like you, so.
Oh, well, no, there are lots that don't smell very interesting, and then you get one that's just shockingly, strongly smell. Like my favorite was the one that smelled like freshly cut grass. And you know, I think it's actually for a lot of sponges, it's a good indicator of certain species.
Seems like it should be on the other side back.
I would have never thought to sniff sponges.
I don't know how it happened because I want to say I didn't think about it either. But I love that you did it. And
we
also thank you for processing those sponges last night.
Yeah, it was fun processing the sponges last night. We had a lot to process last night. It was a good, I think it took us about 4 hours to get things finished up. Maybe 3, but it wasn't too bad. The 3 of us worked really well together, me, Steve, and Mary, so it went well.
Have you worked together before? I haven't heard about the science team on there this trip.
No, yeah, I haven't worked with Steve and Mary before, but it's been pleasant. It's been great.
Yeah, that's fantastic. I know we have a list of samples and targets for this dive too, so in about 24 hours, we should be ready to go again.
Cool. Exciting.
Taylor Ann, are you also writing the observations in the science chat? Is that your role, or who does that?
Sometimes, so like normally if we're doing transects or different information that I think the scientists want to know personally to enter on some of their data sheets that they have at home, I'll put that in the chat. But mostly I'm just typing the observations into the event log we have, and then those eventually get posted online in our dive reports and stuff. So.
I see. Got it.
You can also see Taylor Ann's notes live on the Nautilus Lab website next to the video feed, along with the
Really?
Hercules depth and Argus depth. Yeah. So some new data
I
up
did
there
not know
this
that.
year. Yep.
I'm going there. I want to see. Oh, that's fantastic. Ah, that's awesome. Okay.
Hmm. Let's see. And let me refresh it. I'm not seeing NA on.
No, it's reading okay.
Yeah.
34.4.
Can you refresh yours? Huh. That's interesting.
Also, Amanda, you're off SPL right now. I'm not sure if you're aware. Okay.
Hello, I'm back on SPL.
What are you up to at home on watch?
Oh, me? Oh
Yeah.
my goodness. I am here. I've got ROV dive in front of me, and I've got my kitten Marbles sleeping next to me. Yeah, life is good. And we had class today. I teach a scientific writing class, and we were talking about, you know, how to construct good sentences, and I was making their homework today. And I put in a couple things that were definitely ROV dive related. I think Davidson Seamount was on my mind.
Oh, that's awesome. That's really cool.
You know,
that sounds great.
change this sentence into the active voice, and it was about octopuses eating their
Nice.
eggs.
Yeah, I mean, that makes it a little easier if you're writing about something that's cool.
Yeah. Oh my gosh, totally.
Awesome.
Well, how is it? Are you on the ship? Yes, you must be because you're
Yes.
processing samples.
Yeah, I am. It's been good. Been a lot more like I guess work because there's less people. But I don't know, it's still just as fun as last year. We had to go into quarantine for 2 weeks before we could come aboard the ship. So we were all individually in our own rooms for quarantine for 2 weeks, separated from society. So that was kind of interesting, but it was good. And then now we're here and things feel somewhat normal, but you know, we all have masks on and stay distant from one another as much as possible. But yeah, it's good. Processing samples is still my favorite part, of course.
Of course.
How late were you guys up last night processing those samples?
Yeah, we were up till, I think I finished at 4.
Wow.
Maybe 3:45, something like that. Not too bad for all the samples that we had. We had some big coral colonies.
Oh, for the ones that are being aged?
Yes, some bamboos, or we got a Cibogorgia, and I cannot say that one. Steve, do you know how to say it? The
Keratoisis.
Keratoisis. Yes, I can't say that one.
Me neither.
Yeah.
Cool. Oh, that's great. My favorite is Isidella.
Oh yeah, that's pretty.
I know we saw some yesterday, but just the sweeper tentacles that form, and I think they're really interesting. I would love to stare at an Isidella or revisit an Isidella and find out if or how the extent of those sweeper tentacles changes over time, because I think they're just regular polyps that turn into them. Maybe it's induced by a predator coming about and you know, kind of a defense that kind of spurs up once needed.
That's interesting. Yeah, I'm always impressed by what we see, especially when we see corals and sponges. Anything down here is impressing, honestly.
So I'm curious, I think it was, I don't know if it was you, Dan, who said it. Someone yesterday said they've collected a lot of the soulless cucumbers in their life. Like, what do people do with them? What are people studying with them? Do you know?
Yeah, that was me, Steve here.
Oh, that
I'm
was
just
you.
filling in for
Hey, Steve.
for Chad while he's taking dinner. Yeah, it's strange, you know, the respiratory trees kind of look like corals, so a lot of times people sample them thinking that they're corals. But, you know, once they retract in, you know, they just can look like a potato.
Right.
So, yeah, I don't know, no one's really requested them, but yeah, they're just kind of accidental sampling, and they're super abundant in different parts
They
of the
are.
West Coast. So if anyone wants them, or, you know, send me send me an email. And I have at least 3 or 4 in the lab.
Oh, wow. Well, yeah, I mean, I was thinking about it. I also hadn't really thought of any. I'm not a cucumber person per se, but they were super abundant there in Pioneer Canyon last night. And I just thought, you know, here I am focusing on sponges and saying they're important for their suspension feeding, and really there's also this whole category of sea cucumbers snuffling out with their respiratory trees in the breeze, or, you know, just kind of feeding. Are those the respiratory trees? Or is that the, actually that might be the mouth. I really don't know. I have to look.
Yeah, I'm not sure. I'm not a sea cucumber expert. It's just that was the part that made sense to me.
I'm definitely going to look that up right now. I'm super curious. I'll let you know what I find. Well, the first interesting thing I've learned from looking them up in a paper is that they were originally described by a scientist whose last name was Ekman. Ooh, I'm impressed. Ekman is famous in oceanography for Ekman transport. I did not know that Ekman may have studied biological things as well. But this one does not say where the front end is, where the mouth is. So I'm still hunting.
I think they're different Ekmans. They're actually, there's a physical oceanography Ekman, and then there's a zoologist Ekman.
Oh, no way. Well, okay, that makes a lot of sense. I was going to say otherwise, what an amazing generalist. I would have figured Ekman transport was a pretty difficult concept to come up with.
Yeah, I don't know. I think one's an oceanographer, one's a zoologist, but they're both Swedish. And I don't know if they're related. Lived about the same time.
Very interesting. Do you know, maybe they were siblings or family, or maybe they were married? Though probably few women engaged in oceanography or marine science back then. All right, my silence was because I was taking a look at this paper about feeding in Solus, and this one is actually a shallow species of Solus. This is from Solus chitinoides, which is found in snorkelable and scuba diveable depths up in British Columbia. But it is a Solus, and it has a very similar morphology. So for this one, yeah, those tentacles are the feeding tentacles, which means that the hole that they retract to is the mouth. So there's a series of muscles that can kind of pull on those tentacles or the stalk of them, and that can help direct them into the currents. And the other neat thing is that those tentacles have buds at the ends of each of the sort of branching tips. And those buds have a bunch of folds in the tissue. And the folds are there because then you can imagine when you stretch a fold out, it becomes flat. But then when you unstretch it, that fold pinches whatever was there in between it. And that's actually how at least this Solus captures food, is it would stretch out those buds so that they'd be unwrinkled. and then they would contract them again, and that would cause all the wrinkles to come back that would sort of catch whatever food particles were drifting by. And then they would take each tentacle in turn and put it into the mouth and clean off what was on those buds. That is how Solus feeds. Where is the respiratory tree and the cloaca? I'm not sure. I have a feeling it's underneath the armored surface, but I haven't found that written out yet.
Yeah, that would make sense. The underside is actually quite smooth. So if you manage to pry them off a rock, the underside is kind of this translucent. You can see the internal anatomy pretty clearly.
Interesting.
Yeah.
Oh, that's cool.
I have some photos I can send you if you're curious. We tried to dissect one last year, yeah, from off Catalina, a deep one.
That would be great. I'd love that.
Mm-hmm.
Here, I'll send you my email address in the chat.
Sure. Yeah, I was a little worried that when they close up, I was a little worried that the preservative, the ethanol, would not get in there. So we ended up injecting a little bit of ethanol into the underside. It seems to be more fleshy, and we were able to preserve it that way.
That sounds like that would work pretty well. So that did work?
Yeah, it did work.
Oh, cool.
I get the feeling that since they're so abundant, people tend to overlook them as something that's known, but I don't really know much about them myself.
Yeah. Well, I agree. I mean, I remember making the exact mistake that you mentioned of the first time I saw them, I thought they were some tiny coral. And then I was blown away when I learned that they were a cucumber. And then I did a lot of research up in British Columbia at Banfield Marine Station, where this shallow species, Solus chitinoides, lives. So I remember checking out those Solus when we would go out tide pooling and snorkeling. I don't know why I didn't really take a stronger interest right away in them, because they are actually pretty interesting. Just sort of how they're built. They're so weird. The armored plates are really cool. And they're probably, they have such strong bilateral symmetry, which is not typical for echinoderms. I mean, yes, all cucumbers have fairly bilateral symmetry compared to their relatives, but these guys are even further down that path. Now I'm on a hunt looking at Solus papers. Okay, how are we doing descending? Oh, 2,190 meters. We are getting close. Sort of, 1,200 meters more. Oh, we're going past an octopus right now. I'm sorry, a squid. You can see it there, descending faster than it is. So for those keeping track of our descent, we're at 2,360 meters depth, and the oxygen concentration has crept back up now that we're past the oxygen minimum zone. So now the oxygen saturation is 16%, but it's getting colder. Right now, the water temperature outside of Herc is 1.9 degrees Celsius. We've been passing by several larvation houses. So I know last night we saw a lot of them on the ascent as well. We were talking about them. But larvation houses are neat. They're these mucus houses that are sort of inflated with water by these little tadpole-like animals called larvations. And so the larvation actually has special cells on the top of its tadpole head called oikoblasts, which make this cool mucus mesh. And then once that mucus mesh is ready to go, the larvation will pump its tadpole-like tail to fill up that mucus house and inflate it with water. And the tadpole, well, it's not actually a tadpole, but it's tadpole shaped. The larvation sits inside of that house and pumps its tail to help draw water through that whole mucus structure. And that structure turns into a series of tubes and canals and filters. And so this is a way that larvations filter feed from the water. When we see the larvation houses here, it's hard to know whether they are occupied or not right away. Although I'm actually seeing some of them that you can tell if they're unoccupied. They're sort of shaped like parachutes, where there's sort of a heavier end and then it flares out to a rounded end at the top. That's probably a discarded larvation house. And that's the thing, larvations will produce a new mucus house every day, sometimes even more frequently than that. So as they filter, that mucus gets covered in particles and sediments and whatever else kind of drifts past. And eventually they become clogged enough that the larvation itself will sort of swim out of that house and just let it sink to the seafloor. These mucus houses have been quite important in terms of transporting carbon into the deep sea. So they're big, and that mucus captures particles and then moves them down quickly to the deep sea floor. So it's a way that there's a large amount of carbon from shallower water that quickly gets transported down to depth. And why being quick matters is because the longer something drifts or sinks through the water column, the longer that all of the critters that you see here, and some of the critters that you don't see, like bacteria and protists, can start to nibble away and eat the carbon that's sinking down. And because of that, the deeper it is, the less food arrives at the seafloor. So it's estimated that for every 1,000 meters of depth you go, you only have about 10% of the food that was at the surface or at the previous 1,000 meters. So here we're going down to 3,000 meters, so where there should be less than 1 or even 0.1% of the food that's currently there. I think we typically estimate about 1 to 3% is what gets down to about 3,000 meters. Just to check on the Grafana output for the oxygen optode wand, there are 3 plots in a row: optode wand temperature, and then optode wand saturation uncorrected, and then the wand concentration uncorrected. And then there's another plot below that that has oxygen saturation and concentration. It says corrected. Is that sorry, my connection's going out a little bit. Is that from the optode as well, that one that's corrected?
Hey, Amanda, I got an answer in here. So that fourth one is actually the built-in optode on the vehicle itself. So that has a correction factor on it.
I see. So we're gonna want to correct the optode later with the temperature that you get and the salinity?
Yeah. So there was an email sent out today to Jeff and his crew about getting that correction value that could have been plugged in, but it can be done later, I guess. Yes, I'm getting a nod. It can be.
And sorry, I keep cutting in and out of the V-Link chat right now, so I'll talk when I can.
Oh, no worries. If you didn't hear that, those values, there was a request to get the corrected or correction numbers today, but it was kind of last minute notice, but those can be applied later.
Okay, that's great.